everyone and welcome to Instant Biology by Dr. Neelab. The topic that I'm going to teach today is lysozymes. Now you might have heard about lysozymes, and basically it is a very important tool, or it is very important enzyme in case of the recombinant DNA technology. So when we study recombinant DNA technology, you might have seen that I've already made a series on RDT in which I have discussed about multiple enzymes, and I've also discussed about lysozyme, but that was in Hindi. In this current video, I'm going to talk about lysozymes, but that would be in English. So we would be talking about the mechanism of lysozyme, how it works. We would be talking about where the lysozymes are found, basically sources of lysozyme. And we would also be talking about the applications of lysozyme, where lysozymes are used. So please be with me in this video till the last. You would be definitely gaining a lot of things that you might not know up till now. So let us start. What are lysozymes? Lysozymes are enzymes catalyzing the hydrolysis of beta-1,4 glycosidic linkages. Now what do you think beta-1,4 glycosidic linkages are? Now beta-1,4 glycosidic linkages are the linkages that basically link up two carbohydrate moieties. Now suppose this is one carbohydrate, this is another carbohydrate. When they are coming close to each other, they can form a bond. One such bond is beta-1,4 glycosidic bond. So glycosidic bond means that it is between the two such residues that are carbohydrate moieties. Okay. Now what do you mean by, we understood what is glycosidic, right? But what do you mean by 1,4 glycosidic bond? Now understand that beta-1,4 glycosidic bond means beta stands for the positioning of the OH atom. Okay, of the first. Now suppose this is the first carbohydrate residue, and in this carbohydrate residue, the anomeric carbon has a OH group placed above. It is positioned above, then it is called as beta. If it is positioned downwards, then it will be called as alpha. So in case of lysozymes, it is able to cleave beta. Okay, now let us talk about 1,4 glycosidic linkages. What do you mean by 1,4? Now suppose the first carbon is taking part in the reaction and the fourth carbon is taking, I mean to say is the bonding is between the first carbon and the fourth carbon. First carbon is of the first carbohydrate moiety, and the fourth carbon is of the second carbohydrate moiety. I'll be showing you that in a diagram in just a bit. So that is the bond, right? So 1,4. That means this is the first carbon that is forming a bond with the fourth carbon, and this bond is called as beta-1,4 glycosidic linkage. And what does lysozyme do? Lysozyme is an enzyme that cleaves off the beta-1,4 glycosidic linkage. So I hope that would be clear in your mind. Now let us move on to the next point. Where is this beta-1,4 glycosidic bond majorly present? Now this beta-1,4 glycosidic bond is most commonly present in case of the cell wall of the bacteria. Now bacterial cell wall. Now what is bacterial cell wall basically made up of? You might be knowing that bacterial cell wall is actually made up of peptidoglycan. What do you mean by peptidoglycan? Now if I talk about peptidoglycan, let me just write it down over here. Peptidoglycan basically comes from two words, peptido and glycan, right? What do you mean by peptido? Peptido means the peptide linkage that is present between the two NAG-NAM chains. Okay. Now there are suppose there is one chain that is composed of NAG-NAM, NAG-NAM, NAG-NAM. What do you mean by NAG-NAM? Basically these are it is the composition N-acetyl glucosamine and N-acetyl muraminic acid. These are the two components of the peptidoglycan. Okay, so one is NAG, one is NAM. So when we mean glycan, when we say glycan, what do I mean by glycan? I mean by glycan that there is a NAG and there is a NAM. And suppose this is one chain of NAG, NAM, NAG, NAM, and this is another chain of NAG, NAM, NAG, NAM. And these two chains are joined together by peptide linkages. That is why this particular complete thing is called as peptidoglycan. I hope you would have got this clear in your mind. If not, I'll be showing you a diagrammatic representation, and then it will be clear in your mind that what is peptidoglycan and how does it work. Okay. Now let us move forward. So NAG-NAM are the components of the peptidoglycan cell wall of the bacteria, right? So it is present in the cell wall of the bacteria. Now Animals do not have cell wall, right? Plants have cell wall and the microbes have cell wall. Plants cell wall is made up of cellulose. Okay, therefore lysozyme, I mean basically it is made up of cellulose. But over here, in case of the bacteria, the cell wall is made up of peptidoglycan. And peptidoglycan means NAG-NAM and N-acetyl glucosamine and N-acetyl muraminic acid. This is the full form. Let me just underline it over here. N-acetyl muraminic acid, this is called as NAM, and N-acetyl glucosamine, this is called as NAG. Now let us move forward on to the next slide. In this slide, it will be much more clear that how does lysozyme function. Now you can see that this, for instance, is, just a minute, I will take up a pen. This, for instance, is NAG and this, for instance, is NAM. The purple color is of NAG and the orange one is of NAM. So this is, you can see over here NAG and this is over here NAM. Now you can see that NAG, NAM, NAG, NAM, this is one chain and this is another chain, NAG, NAM, NAG, NAM. And they are linked up with the peptide bonds over here that are present over here. What does lysozyme do? Lysozymes will come and cleave off this beta-1,4 glycosidic linkage. So this is beta-1,4, that means the NAG and the NAM are linked up with the beta-1,4 glycosidic linkage. To be more clear, over here you can see that this is the structure of NAG. Okay, it is a carbohydrate. Look at this. This is again NAM. What is the bonding over here? The bonding is the first carbon and this is the fourth carbon. So the bond that is present over here is beta-1,4 glycosidic linkage. Now why am I calling it beta? Beta I am calling it because the OH position is over here. It is for the first carbohydrate moiety, the OH is positioned upwards. That is why I am calling it beta. If it would have been downwards like this, then I would have called it alpha, alpha 1,4, alpha whatever. I believe you would have got a clear understanding about this, that what does lysozyme do, and what it is doing is it is cleaving this. You can see that the beta-1,4 glycosidic bond is cleaved by lysozyme. This is how lysozyme basically functions. Okay, I believe you would have got a clear understanding about the lysozyme's function. What does it do? How does it do? Let us move forward on to the next slide, that lysozymes can lyse bacterial cells. What do we mean by this? That since now you understand this, that since the bacterial cell wall would be broken down, okay, the bacterial cell wall would be broken down. If the bacterial cell wall is broken down, automatically the bacterial cell would lyse, okay. This is what is written over here, lysis of the bacterial cells. Now, present abundantly in, where is it located? Now we are talking about the source. Source of what? Source of lysozyme. What is the source of lysozyme? Lysozymes are present in tears, saliva and mucus. Now basically you might understand this, that tears, saliva or mucus, basically these are the secretory things, these are the fluids, the body, right? So basically lysozymes would also serve as something that would be protecting your body, right? If they are coming out from the tears, whatever bacteria is attached onto your skin, or if it is coming out from your saliva, whatever food that you are ingesting, if it is having any kind of a pathogen or it is any kind of a bacteria, it might be broken down with the action of the lysozyme. This is what the lysozyme's actual function it is trying to protect your body from any kind of a pathogen. Okay. Now let us move forward. Applications. Lysozyme is used for cell wall lysis in the cosmid and in the plasmid DNA isolation procedure. Now you might be knowing that bacteria are having, some bacteria, not all, some bacteria are having the extra chromosomal genetic material that is called as plasmid. And most microbiologists, they want to isolate plasmid for the different functionalities, for the different utilities. If you have to isolate the plasmid, what you have to do, first of all, you will have to break down the cell wall of the bacteria. Now again, the primary way or the best way to break down the cell wall is this lysozyme. You can break it down with the help of lysozyme. However, there are other things also that you can take help from, but lysozyme can be one such method in which you can break down the cell wall and then isolate the plasmid. Okay, so this was the functionality, this was the utility of lysozymes. I hope you would have liked the video. If you liked the video, please hit the like button and comment also on the video that what did you like about this. And with this I would be making more such videos in English, so that all my audience are benefited from this video. Again, please do subscribe the channel and share it with your friends. Thank you so much. Have a good day.